Good morning. I'm Ed Bertschinger from MIT, and today we'll give my second lecture on cosmic evolution, focusing on the formation and evolution of planets and life. You remember that in the first lecture, I began with an opening image of the sky, one that I had seen with my own eyes. This is a view of our Milky Way galaxy, of a small portion of our galaxy, taken with the Hubble Space Telescope. It shows a cluster of brilliant, massive young stars formed in the deep of space, surrounded by the birth cloud of gas and dust, mostly hydrogen gas, from which those stars were formed. The bright light of the stars is blasting away, pushing away the clouds of gas and revealing some pillars or fingers of dense gas where new stars themselves are being born, with the winds of those heavy stars blowing past them. These are stellar nurseries, the birthplaces of stars. Images like this raise questions that I asked at the beginning of my lecture on Monday. What is this beauty in the heavens? Where did it come from? Are we part of it? Are we of the same stuff? Are we of the same laws as that in the universe? And where did we come from? Last time I also introduced this slide of sizes of objects in the universe, ranging for the universe itself, the part that we can see, the visible universe, 10 to the 24 meters. Our galaxy, 10 to the 21 meters. Stars, like the sun, 10 to the 9th meters. Planets, and then down at the human scale, people. This time, I'm going to also consider objects that maybe are even smaller than people, the origins of life. And as we extend down to the smallest scales of nature studied so far, we get to the elementary particles, such as protons, from which all matter that we know is made of. Now, last time I focused on the largest objects, the galaxies, the universe, and stars. They are simple, relatively, compared with objects like people or planets. And similarly, the elementary particles, such as protons and even the molecules that are composed of atoms, are relatively simple. It often happens in nature that extremes are simple. It's the moderates who are complicated. The simple objects are those that are amenable to laws of physics that have predictive power, that we can calculate and understand in some detail. And so physicists and astrophysicists study these. We do it because we're simple people and they're simple objects. Today I'm going to focus on the more complex planets and human life of various kinds, which is very complicated because so many parts interact and so much is dependent on the history of interactions. These are the realms where the laws are harder to discern and where instead of constructing many grand theories, we use a few principles and lots of observations. So I will begin with planets. And I'm hoping that the sound can be turned up just a little bit for this video.
The places where stars and planets are born are among the galaxy's most beautiful locales. These cosmic landscapes change as new generations of stars light up and disperse their birth cloud. But the youngest stars seen here are already perhaps 1 million years old. Hardly toddlers.
Stars and planets form in the dark, inside vast cold clouds of gas and dust, such as these pillars imaged by the Hubble Space Telescope.
The dust is so thick we can't see the infant stars inside, at least not with visible light.
With infrared light, Hubble can see through all but the thickest dust. Yet it's in those dense knots that the youngest stars are forming.
To peer inside them, astronomers need the James Webb Space Telescope.
With a mirror larger than Hubble's and performance optimized for the infrared, Webb will give astronomers their closest look yet at stellar birth.
We're flying through a computer model that represents astronomers' best ideas about the star formation process.
Reddish colors indicate thicker dust. The temperature, less than 400 degrees below 0 Fahrenheit.
Or less than 240 degrees below 0 Celsius.
That pinwheel ahead is a protostar, perhaps 10,000 years old. Protostars arise when a dense knot of dust, less than a light year across, collapses. But the details of the process are not well known.
Elsewhere in the cloud, another protostar is preparing to build planets. As the cloud that created the protostar collapsed, it flattened into a disk. The disk we see here is 600 times the size of Earth's orbit around the Sun.
If placed in our solar system, it would extend far beyond the planets.
In this computer model, the disk continues to accumulate gas and dust from its surroundings for thousands of years. Eventually, the disk fragments, producing dense, bright structures. These may become sites where giant planets form.
Later, during another phase of construction, smaller Earth-sized planets may take shape.
At least, that's what scientists think happens. It will take the Webb telescope's keen infrared eye to see what's really going on in the cold heart of stellar nurseries.
The Webb Space Telescope will be launched in 2018, a successor to the Hubble Space Telescope, and it will be extremely sensitive to the measurements of infrared radiation that can penetrate through the dense birth clouds where stars are born. That telescope will give us some of our finest views and improve our understanding of how stars are born. Even though we don't understand fully the formation of stars and their planets, we have been able to search for them, and remarkably, in the last 25 years, have discovered a great many planets outside of our own solar system. This work has been aided by a space telescope called Kepler. Kepler was a German astronomer who lived 400 years ago and who observed the motion of the planets around our sun, thereby establishing the basis for Newton's theory of gravity. In honor of Kepler, NASA named its satellite, which is exploring the heavens, searching for planets around other stars, other solar systems, using, in fact, Kepler's same kinds of observations to deduce properties of those planets. The way that the spacecraft works is not by viewing the planets directly. They're too small, far too small, and their host stars too bright for us to see the planets. Instead, Kepler uses the same ideas that apply in a solar eclipse on the Earth. When the moon is properly aligned with the sun, the light of the sun is darkened. Similarly, when a planet around a distant star is properly aligned in front of that star, the light of the star will dim. Perhaps not completely, because the eclipse usually is not total, but in part. And so, if a planet passes in front of the star, the light of the star will dim slightly, and then as the planet exits the face of the star, the light will increase again. And you can infer that the amount by which the light of the star decreases tells you how big the planet is compared to the star. And the duration, the length of time for this diminution of light tells how big the planet is. And also, by watching repeated eclipses, we can determine the orbital period of the planet. So we can measure the size of the planet, we can measure the orbital period, and combining those, we can measure the mass, and knowing the mass of the star, we can compute the radius of the planet's orbit. And that tells us a lot of information. Knowing the mass of the planet, the size of the planet, the radius of its orbit, While we cannot see the individual planets, we can in fact deduce some of their important properties. For example, the density. We can determine whether a planet is composed of heavy elements that make rock or lighter elements like hydrogen and helium, gases. In our own solar system, the inner planets are rocky and heavy. The outer gas giant planets, Jupiter and Saturn, are very low density by comparison. In fact, if we had an ocean large enough, Saturn would float. Remarkably, the Kepler Space Telescope has allowed us to discover not only hundreds of planetary systems and more than 1,000 planets, but among them, at least 30 which orbit their host stars at the right distance where life, as we know it, could exist. These are planets in the so-called habitable zone where liquid water could exist on the surfaces of the planets if they had a sufficiently dense atmosphere. That is a remarkable change in our understanding of our universe in the last decade. As an example, one of the star systems observed by Kepler, around a star named Kepler 62, was found not to have just one planet crossing the face of the star, but 5 of them. Can you imagine five kinds of moons, 5 objects in our own solar system crossing the face of the sun? In fact, something like this does happen in our inner solar system because we have 2 small planets, Venus and Mercury, which do cross the face of the sun occasionally. Those are events called transits. And a famous transit of Venus was used to measure the size of the solar system more than a century ago. And just a few years ago, Venus again transited the face of the sun, a rare event, actually twice in a human lifetime. Venus transited twice in the last 10 or 15 years. The measurements of these planets have told us that 2 of them, numbers E and F, lay at distances around that star where the temperatures are comparable to those for Earth or Mars in our solar system. We would love to know, is there life on those planets? How will we find out? It's very difficult using our technology or any conceivable technology to make images of those distant planets that would allow us directly to see the life in the same way that we can use satellites in orbit around the Earth to peer down and see life here. But what we could do is to look for traces of life in the atmospheres of those planets, molecules associated with life, so-called biosignatures. And there is hope that astronomers will be able to search with future space telescopes, not the Kepler, but the next generation beyond, perhaps 10 years following. And so perhaps in our lifetimes, we will have the possibility and maybe even the actual detection of the molecules of life elsewhere in the universe. What have we learned about planets? Well, in summary, our Milky Way galaxy is teeming with planets. Kepler has found more than 1,000. We know in total nearly 2,000. And just by extrapolating from the rate of discovery and the amount that we've searched, we've concluded that the Milky Way galaxy contains more planets than stars. And it contains, as you know, billions and billions of stars. We know from some of these planetary systems that they are arranged differently than our own. In our solar system, the rocky planets are close to the star and the gas giant planets are far from the star. We know of some star systems where the gas giant planets are very close to their stars, too close to have formed there. And so there are some interesting puzzles about how those planetary systems have evolved, which astronomers are working to understand. We have found many planets that could have liquid water, and our task in the next decade is to confirm whether they in fact have water by looking for water molecules in their atmospheres. We know that planetary systems are still forming in our galaxy, and we'll use the James Webb Telescope and others to learn more about them. But there are key ideas that we've not been able to fully understand or pursue. We don't know really what the planets look like and whether any contain life. That's work for future astronomers. We also know a great deal about one planet, our own, Earth. And I'm going to spend a little bit of time talking about Earth, its geologic history, and life on Earth. How do we learn about the past? Astronomers learn about the past by viewing light that was emitted in the past. Distant galaxies, the light from distant galaxies has traveled a long time to reach us. And so by looking for the most distant galaxies, that's how the Hubble telescope has allowed us to look back in time to the earliest moments of galaxy formation, not of our own Earth, but of distant galaxies. However, we can't use this approach to learn about our own past or the past of the Earth. So instead of looking for light that came from distant places, we look at the Earth itself. This is the Grand Canyon in Arizona in southwestern United States. A river has cut a channel through rocks. And at this place and many others, we've discovered layers upon layers of rocks, many of those layers containing fossilized remnants of animals and plants. And like an onion that grows by adding layers, our Earth's crust is constantly changing with the addition of layers. A sea floor will deposit sediments, such as the calcium from the mollusks and the bones of the fishes. When it rains on the Earth, then the mud that comes down from the mountains is deposited as layers. We, of course, see these layers in our ancient cities, and we dig in them to find about the past of our civilization. By digging through many meters of rock, geologists have learned about the past history of the Earth. What they have found in the Grand Canyon is there are several sets of rocks that measure different times and different sequences of events. Geologists are able to determine the ages of some of those layers by using the properties of certain chemical elements called radioactive nuclides. There is a form of the atom potassium called potassium-40, that's the symbol K40, that is unstable and after about 1 billion years will change to argon. Argon is a gas. We have this calcium-40 in our bodies. It's in our bones. It's in our skin. Not much, but a little. And occasionally, one of those potassium atoms in our body changes to an argon atom. Argon is a light gas, and it just escapes our body. But suppose that we were trapped underground. Suppose that we look at the potassium that is contained in the rocks themselves. The argon released there by radioactive decay would be trapped in the rocks. And so by looking at rocks that have formed, for example, molten rock which from volcanoes, which then solidifies, cools and solidifies, we can find the moment when those rocks were able to begin trapping the argon by measuring the amounts using precise measurements of the rock. That's called radioactive dating. And it has allowed us to assign ages to these different layers of rock. And these kinds of layers are associated with different fossils around the world. There are many patterns that repeat. And so by this process of looking at the layers of the Earth and using radioactive decay, since the 1950s, geologists have been able to construct a geologic history, in this case of the Grand Canyon and elsewhere around the Earth. The fossils are very important because they're remnants of life cast in stone. Among the oldest well-known ones are these strange, almost fish-like objects that lived in ancient seas called trilobites, 3 lobes from their appearance. They lived between about almost 600 million years ago, 500 million years ago, until about 250 million years ago. And here's an example of a fossil trilobite from Canada. We learned that 95 million years ago, before the present, in Palestine, there was a snake with legs, like a lizard. That snake is extinct. There are no snakes with legs present on the earth today. We've learned that there were objects very similar to modern-day whales, the largest mammals living in the sea, but whose nostril, through which they get air, instead of being above the eyes today, was forward in the skull and at the end almost of their snout, like that of a dog or another mammal 50 million years ago. We learn in school, many of us, about these layers and the names that geologists have assigned to the different layers of rocks, and we learn about the types of fossils that were present there. And we learn about major events of the Earth taking place around the world. What these show us is that there were different forms of life on Earth in the past. The forms of life have evolved. And as always, that encourages us to ask, how? Charles Darwin was a naturalist in the 19th century who studied life, fossils and living animals around the world, especially around South America, in his famous voyage as a young scientist aboard the HMS Beagle. He found patterns that puzzled him, and he thought about them for many years. In a set of islands off the northwest coast of South America, the Galapagos Islands, he discovered a set of birds. Of course, they were known to the people living there, but he was the first European to study them in any detail. And he found that the birds were all variations of the same kind of bird, but they differed in some essential ways. The birds living on different islands had different beaks. These islands were widely separated so that the birds did not travel between them. Darwin went to those islands and found that the shape of the beak was formed appropriately to the needs of the bird for survival. On islands in which the birds ate insects, the beak was skinny and probing so that the bird could seize the insects, grubs or insects. Where the bird had to eat fruit or nuts, the beak was a strong tool, like heavy pliers, for cracking the seeds or opening them. For islands where the birds would extract nectar from flowers, the finch beak allowed it to hold a stick and reach into the flowers and probe with them. This was so remarkable because Darwin realized that these birds, even though they had different forms, appearances, and uses of their beaks, were all basically the same. And so he began to speculate, as you see in a very famous drawing. He says, I think that perhaps these different birds were related. Perhaps their ancestors all came from one finch, which then spread across the islands. And over millions of years, the birds changed in order to survive on the islands where they found themselves. This is Darwin's great idea, evolution by natural selection. Natural selection sometimes being stated as the survival of the fittest. If you have a population of birds with the occasional variation or mutation, change in the bird just by chance at birth, then those birds, those animals, those creatures which are better able to survive in an environment will survive and pass along their properties to their sons and daughters. Darwin even speculated that all species of life are related and evolved from a common ancestor. Darwin's theory was incomplete. He didn't know how information about the form and function of life was passed from one generation to another. He didn't know what information that was. How is it that a human knows to develop 2 arms and 2 legs and to walk upright? How is that information passed through the single cell that is the beginning of life? And how can that information change so as to evolve new species? The answers to those questions have to do with genetics. Soon after Darwin's work, an Austrian monk named Gregor Mendel bred the common pea plant. And he found that there were different color varieties of the peas with different properties, that when he fertilized them in different ways, could generate these varieties. And through his studies, he was able to show that the individuals, in his case the plants, the individual plants, inherited form and function in discrete units today called genes. If Mendel had made this discovery 50 or 60 years later, perhaps we would call these quantum theory of discreteness, in this case the discreteness of genetic information. Now, the cause of those genes, their physical nature, was not understood. I've said that life is complex. Biological systems are complex. We are unable to develop theories starting from the laws of physics, which can tell us why life is the way it is. So we have to study it in detail. And this means looking at the molecules of life contained in the nucleus of cells. In the early 1950s, there was a group in the United Kingdom who was studying the structure of the molecules of life. And one of the most talented scientists pursuing this was Rosalind Franklin, a woman working in Cambridge, who was able to extract the DNA molecules from nucleus of living cells in the form called chromatin of the chromosomes, and to crystallize it sufficiently to use a method called X-ray scattering or X-ray diffraction to produce information, to give information about the shapes of those molecules, the DNA molecule. She was working with an advisor who told 2 visitors, Francis Crick and James Watson, about her investigations and gave them some of her preliminary data. They published it, unraveling the structure of the double helix of DNA. For that, they and Franklin's advisor, Maurice Wilkins, won the Nobel Prize in 1962. Sadly, Franklin died of ovarian cancer several years prior. Many people now think that she was the true hero for the discovery of DNA, but she died before she could receive the appropriate credit. You may know that this molecule consists of 2 spirals, certain handedness or parity, joined together by one of 4 light molecules, adenine, thymine, guanine, cytosine, in what are called base pairs. They're like ladders, rungs of a ladder. And these molecules form together in a way that give us a code, a sequence of these letters, ATGC, ATGC, GCAT, and so on. Almost like the binary code that our computers use today, composed of these base pairs. Our genes are sequences of DNA in which these base pairs write out the computer instructions for our cells to produce the proteins that make up us and all life. Now, these are wonderful ideas, and certainly we've learned much about the molecules, but is this story really believable? How can we know that evolution actually takes place? You may see it when you believe it, or you may believe it when you see it. Take your pick. Here's a beautiful example of evolution in practice. seen in the Sonoran Desert of southwestern United States and northern Mexico. There is a small mouse that lives in the desert called a rock pocket mouse. And in most of the desert, it's covered with sand, and the mice have this light color, sandy color. But in some regions of the desert, about 1,000 years ago, or in some cases longer ago, lava flows covered sections of the desert with black, dark rock, such as this dark rock. Now, a light-colored mouse on a dark rock would be easy prey to an owl, a hawk, or something it likes to discover and catch and eat small animals. But a dark-colored mouse on the same black rock would be very hard to see. In fact, you may not even be able to see the mouse in this photograph, and you only can see its tail. And conversely, that same black mouse on the sand would be easy prey for an owl or a hawk. Well, so what? What does that prove? If we look a little more closely at this situation, we can study the genes and the DNA of these mice and learn something about how they are related, the dark mice and the light mice. And we can even learn approximately when they had a common ancestor. What studies have shown is that the DNA of these two kinds of mice that live in the same region, in this case of New Mexico, are almost identical. Gene after gene is identical. But there is one gene, one section of DNA, responsible for the production of a protein called melanin, which gives color to skin or fur. Melanin is why some people are dark-skinned and others are light-skinned. And in the gene that encodes the production of melanin, 4 base pairs out of thousands are different between those two mice. It's remarkable. It's remarkable. The animals are almost genetically identical aside from the gene that distinguishes their fur color. Moreover, the scientists have looked at different lava flows in different places in the Sonoran Desert, and they have found that the mutations are not the same, that there are different ways of changing the melanin gene to turn it on and to cause the fur to be dark, supporting Darwin's idea of the random mutations. The mutations are random, but the natural selection is not. Once you have a mouse with dark fur, it can survive on the dark rocks. Natural selection is very deterministic. If you are visible to a predator, sayonara. And the mathematics works. If only 1% of the mice are dark to begin with, a very rare strain on those dark rocks, and suppose that they are 10% more likely to survive. In fact, the experiments suggest that they're much more than 10% more likely to survive. Simple calculations show that after 100 years, 95% of the mice living on the dark rocks will be dark. That is natural selection. This, of course, raises profound questions. Did humans also evolve? Well, with humans, our genetic codes are more complicated. The gene differences between different humans are much, much more than 4 base pairs. There's considerable variety. And we think the reason is that if humans did have a common ancestor, that ancestor did not originate 1,000 years ago, as in the case of the mice on some of those lava flows. But we can also look at the shape of skulls and the bones of hominids, creatures of the genus Homo, related to Homo sapiens, our species. and learn something from their placement in the fossil record of when those species lived, and by comparing them, their forms, try to reconstruct a tree of evolution, the tree of life, similar to what Darwin did. And as you may know, there are many uncertainties about that. There are some missing links, there are question marks in this diagram where a fossil discovery could help us to understand better the relationships of some of these creatures. One thing we have discovered recently, though, is that there were species other than our own, species of hominids other than human beings, which lived at the same time in the relatively recent past, about 100,000 years ago in the case of the Neanderthals, and maybe even as recently as 15,000 years ago in the case of a species called Floresiensis, which lived in Southeast Asia. Now, newer genetic information uncovered in the last 15 years or so strengthens the case, but this still is not as conclusive as it was for the rock pocket mice. The DNA, which encodes our structure, is present not only in the nucleus of a cell, but also in little parts of the cell called mitochondria, which are the power units of the cell. The DNA in those organelles, as they're called, comes not from the father and the mother, as it does for most of our DNA, but it comes only from the mother, so-called matriarchal DNA. And this DNA, therefore, is preserved more completely with fewer changes, and is easier to discover the genetic relationships among different humans using mitochondrial DNA. A number of years ago, it was discovered that the rate of evolution of random variations of mitochondrial DNA implied that all the humans that have been studied from around the world had a common ancestor who was given the name mitochondrial Eve in the popular media, like the Eve of the Bible. And this common ancestor lived in Africa only about 150,000 years ago. Now, it does not mean that humans originated 150,000 years ago. There were, of course, ancestors before who gave birth to the ancestors of Eve herself. But it is remarkable to think, and consistent with our studies of fossils, that we, all in this room, came from a common ancestor in Africa as recently as 150,000 years ago. What have these studies taught us about the evolution of life on the Earth? First, we found that we can see the past not using telescopes on Earth, but using fossils and geology and the geologic record. We've learned that life forms have changed repeatedly during about 3.5 billion years, the period of time for which we have fossils out of the 4.5 billion year age of the Earth. But one thing that all forms of life have in common is this genetic information encoded in DNA. I will say there are symbionts, there are molecules or structures, objects called viruses, some of which use RNA, a simpler relative of DNA, to encode their information. But they are often classified differently than the cells of life because they don't have the ability to reproduce and thrive without a host. We've learned that evolution saved the rock pocket mouse. Random mutations of its genes, different random mutations in different places, allowed the pocket mouse to develop dark fur and to survive on the lava flows of the southwestern U.S. deserts. We've learned that humans have evolved with other primates during the same time. In fact, we've even learned more. Since the year 2000, biologists have been studying the DNA of humans, deciphering the complete genetic code, in something called the Human Genome Project. In fact, nowadays, you can go on the internet and pay less than $100 to get the complete sequence of your personal genome. Studies like this have revealed further remarkable facts. The DNA is a long molecule, and there's lots of it in the chromosomes of the nucleus of the cell. The studies have shown that less than, in fact, about 1.5% of human DNA codes for the proteins. That is, the code sequence of the ATGC labels the production of protein molecules. Most of the DNA does not encode the production of proteins. It has other functions, and we've come to understand that it's very important for regulating those genes and telling the cell, for example, when to divide, when to grow, when to do different things, when to produce the molecules that make my arm extend. Amazingly, we've discovered that about 8% of human DNA came from viruses. Viruses, for example, those that give us the common cold, or some of the more dangerous viruses like the HIV virus, which produces AIDS, they can modify the DNA of their host organisms and even modify it in a permanent way. 8% of our human DNA approximately has been permanently changed by viruses. About 3% of human DNA came from Neanderthals, from what we thought was a different species that went extinct over 100,000 years ago. Our DNA is mixed up with that of the Neanderthals. Did we murder the Neanderthals, or did we mate with them and more successfully breed than they did? We don't know. We've also found in recent years that humanity has a great responsibility now for life, because we have found that we can change life through genetic engineering. Recent discoveries, in fact, allow gene editing to go in and change the very structure of the DNA molecule at specified points. Therefore, humans are now causing biological evolution. This brings us back to where I began, with perspectives on the universe and our place in it. I ask questions about the beauty of nature, its origin, whether we're part of it, where did we come from? We've answered some of those questions for the universe at the large, for stars, for planets, for life on Earth. Those have given us a great sense of mystery and awe and responsibility as citizens to learn, as scientists to understand, and as societies to use our knowledge wisely. Thank you very much for your attention.